I'm Dr Gemma Sharp, and in this lecture I will be introducing Mendelian randomization, which is a technique that uses genetic data to infer causal relationships in observational epidemiology. By the end of this lecture, you'll be able to describe how MR can help overcome some of the issues that plague observational epidemiology, so confounding, bias and reverse causation, give examples of the use of MR in observational epidemiology, list different varieties of MR and the context in which each could be used, and explain some of the methods that can be used to explore violation of the main assumptions of MR. So we know that randomized control trials are the gold standard study design in terms of causal inference in epidemiology, but sometimes it's not feasible or ethical to conduct an RCT. So since we can't always rely on RCTs, instead we have to rely on observational studies. But inferring causality from observational data can be tricky. Observational studies can sometimes give the wrong answer because of confounding, reverse causation and other types of bias. So for example, results from observational studies have suggested that hormone replacement therapy, or HRT, can prevent cardiovascular disease. Vitamin E supplements can prevent cancer and CVD. And therapeutics aimed at reducing C-reactive protein levels can prevent or treat insulin resistance, type 2 diabetes, and CVD. All of these interpretations are incorrect, and they're the result of misleading associations identified through observational studies. So at best, these can introduce red herrings that mean that research effort is invested in the wrong area, but at worst, they can result in misguided changes to clinical practice and public health advice that are potentially harmful. So one of the issues is that attempts to adjust for confounders at the analysis phase are not always successful. Adjusting for confounders in multivariable analysis doesn't account for unmeasured or residual confounding. It's difficult to measure or think of all the factors that are associated with both an exposure and an outcome. And even if we do adjust for important confounders, if they're not measured correctly, or they're measured with error, or they're modelled incorrectly, then we won't be able to fully adjust for them in regression models. Mendelian randomization is one approach that attempts to circumvent these issues with unmeasured and residual confounding to help infer causality in epidemiological studies. MR is a technique based on the idea that genetics can tell us about non-genetic factors and their effects on health and disease. It uses genetic information as a proxy for non-genetic information, allowing us to test for causal effects in the presence of confounding. And it's about testing causal effects of potentially modifiable risk factors, so things like BMI, smoking, vitamin C, rather than the effects of genes. To understand the principles of Mendelian randomization, it will help to remember some of the basic concepts from the field of genetics. So deoxyribonucleic acid, or DNA, is organised into genes, and DNA encodes the information responsible for the development and the function of an organism. The genome is the term used to refer to all of an individual's DNA. Individuals have slightly different genomes with different genetic variants or alleles, potentially causing different functional effects. Most cells in our bodies have 2 copies of the genome, one inherited from each parent. Which of our parents' copies we inherit is randomly determined when sperm and egg cells are made, and this is in a process called meiosis. What Mendelian randomization does is it takes advantage of these features of genetics by employing them as proxies, or what we term instruments, for exposures of interest in a special type of instrumental variable analysis. So if we consider this example, the research question is, does exposure X, which is a high level of LDL cholesterol, cause outcome Y, which is coronary heart disease? LDL cholesterol is associated with coronary heart disease, but does cholesterol cause coronary heart disease, or is the correlation explained by confounding, labelled here as C for measured confounding and U for unmeasured confounding? Alternatively, is the association explained by reverse causation, i.e. does CHD cause high levels of LDL cholesterol? If X causes Y, so if the exposure causes the outcome, it follows that any factor that influences X will also influence Y. And we call these factors instruments, and here they're labelled as Z. Genetic effects can serve as really excellent instruments. And this is because virtually all traits are at least partially influenced by genetic effects. Genetic variants are inherited from our parents at random and independently from other variants, so they're unlikely to be affected by confounding. In a genetic association, the direction of causation is from the genetic variant to the trait of interest and not vice versa. Genetic variants and their effects are subject to relatively little measurement error or bias. They're measured objectively. The measured genetic variant doesn't have to actually cause the exposure. It could just be in linkage disequilibrium, or LD, with the causal variant, and I'll talk a bit more about LD later in the lecture. And finally, genetic data are now routinely available on large, well-phenotyped studies, which opens the possibility to do more Mendelian randomization studies. So in our example, to address the question of whether LDL cholesterol causes CHD, we could use a randomized control trial. So in a randomized control trial, we would randomly assign participants to either the treatment or the control arm, and this would avoid potential confounding between the intervention and the outcome. In this case, we could randomly assign some people to receive statins, which are commonly used in the treatment of high LDL cholesterol, and some to receive a placebo. If the rate of CHD is lower in the group receiving statins and all other assumptions are met, then we can infer that LDL cholesterol causally affects coronary heart disease. So MR emulates an RCT, but instead of randomly assigning some people to receive statins, in this case some people have been naturally randomly assigned lower or higher on average doses of LDL cholesterol through their genetic makeup, and MR takes advantage of this. So in MR, if a particular genetic variant is robustly related to an exposure, people with that variant are effectively being assigned a higher on average dosage of that exposure, and people without that variant are assigned a lower on average dosage. If the exposure causally affects the outcome, we can therefore expect people with the particular genetic variant to have a higher rate of the outcome than those that did not inherit it. As with an RCT, because the genetic variants are randomly assigned during meiosis, they should be unrelated to confounders. So for example, genetic association studies have shown that the T allele of a single nucleotide polymorphism in the HMGCR gene is robustly associated with lower LDL cholesterol. If the rate of CHD is lower in people with the T allele than in people without the T allele, and if all other assumptions are valid, then we can infer that LDL cholesterol causally affects CHD. MR has been increasingly applied to study the causal effects of a wide range of exposures or traits on a wide range of outcomes and diseases, and there are several good reviews of the use of MR in the literature. MR makes several assumptions that should be met, especially if the research aim is to estimate the precise magnitude of the causal effect of our exposure on our outcome, rather than just inferring whether or not a causal effect exists. Again, we can draw parallels between many of the assumptions of MR and those of RCTs. There are three main assumptions. So the first assumption is that the instrument must be robustly associated with the exposure. So in MR, this means that the genetic variant must be robustly associated with the exposure, and in an RCT, it means that randomization to the treatment must affect levels of the exposure. The second assumption is that the instrument is not associated with any measured or unmeasured confounding factors. So in MR, the genetic variant must be randomized with respect to the confounders, and in an RCT, the intervention must be randomized with respect to confounders. The third assumption is that the instrument is associated with the outcome only via its association with the exposure. In MR, the genetic variant should not affect the outcome via a pathway that does not involve the exposure, and this is known as horizontal pleiotropy. In an RCT, there should be no direct effect of the intervention on the outcome. MR also additionally assumes that all associations, so Z to X, X to Y, Z to Y, and associations with the confounders, are all linear and not subject to interactions. So how likely is it that we'll meet all of these assumptions? Assumption 1, which is that the genetic instrument is robustly associated with the exposure, can be tested through regression analysis and observing the F statistic or the R squared. But the other assumptions can't be checked unequivocally. For example, we can conduct post hoc tests to explore whether the genetic variant is associated with any measured confounders, but residual confounding and unknown confounding is still possible. But if the assumptions are met, then the unbiased estimate of the effect of our exposure on our outcome can be made using two-stage least squares regression, where in the first stage X is regressed on Z, and in the second stage Y is regressed on Z. There are several factors that affect the likelihood that the MR assumptions have been met, and these should be considered when interpreting results from MR. So firstly, MR assumes a robust association between the genetic instrument Z and the exposure X. These associations are identified through genome-wide association studies, but for many exposures and traits, there are no known suitable genetic instruments, perhaps because a GWAS hasn't been conducted. So the solution is to conduct more genome-wide and sequencing-based studies. MR studies often suffer from low statistical power. Genetic variants explain only a small amount of the variance in an exposure or a trait, so power can be low. If the association between Z and X, our instrument and our exposure, is weak, and the sample size is low, then the causal estimate can be biased, and this is known as weak instrument bias. To avoid this, MR studies should use large sample sizes, but they can also combine genetic variants into an instrument that explains more of the variance in X. And another way to increase power is to use two-sample Mendelian randomization, which I'll discuss in more detail towards the end of this lecture. MR doesn't distinguish between a causal effect of an exposure on an outcome and a causal effect in the opposite direction, i.e. reverse causation. However, if both the exposure and the outcome are instrumentable, so i.e. there are suitable genetic variants in existence that can act as instruments for both variables, then MR can be carried out in both directions, and the relative size of the effects can be compared, and this is known as bidirectional MR. Population stratification refers to a situation in which different groups within a sample have different genetic ancestries, and genetic ancestry is correlated with the outcome of interest. This can introduce genetic confounding, thereby violating one of the MR assumptions. The way around this is to use ethnically homogeneous groups and also to adjust for principal components that describe the ancestry of the participants in our sample. Pleiotropy describes a situation where a genetic variant directly influences more than one trait, which raises the possibility that it can be associated with the outcome independently of the exposure of interest. And this would violate the MR assumption that the genetic instrument is associated with the outcome only via its association with the exposure of interest. Pleiotropy is difficult to avoid, but it can be alleviated by using genetic variants that are close to, which is called in cis with, a known causal variant, and also using multiple independent genetic variants because it's unlikely that they would all share the same pleiotropic effect. Some genetic variants, particularly those that occur physically close together on the same chromosome, are highly correlated, and this is a phenomenon known as Linkage Disequilibrium, or LD. LD is crucial in genetic association studies because it allows measured genetic variants to proxy for unmeasured causal variants. To be a valid instrument, the genetic variant or variants used as instruments in MR don't have to causally affect X, provided that the association between the instrument and X arises from LD with a causal variant at the same locus. However, when variants in LD affect the function of other genes, then this can introduce pleiotropy and therefore genetic confounding. One way around this is to conduct studies in populations with different LD structures, and again to use multiple independent cis genetic variants as instruments. Although the genetic variant doesn't have to causally affect X for it to be a valid instrument, it helps to understand the biology behind its association with X. So for example, if a genetic variant is robustly associated with higher levels of circulating levels of protein X, then it's important to know whether this is because it leads to increased production of X, so higher production leads to higher rate of the outcome, or whether it's due to decreased cellular binding of X, so decreased binding leads to lower risk of the outcome. If the genetic instrument is associated with both production and cellular binding, which is known as trait heterogeneity, then this can further undermine the ability to infer causality. Finally, canalization refers to a situation in which during development a functional genetic variant might influence expression of multiple other genes, and this could lead to changes that counter the effect of that variant on an exposure. Again, a better understanding of the biological relationship of the genetic instrument and the exposure can aid interpretation. And it's also worth considering whether the influence of the genetic variant on X occurs during or after development. For example, a variant in ALDH2 is strongly associated with alcohol consumption, but because alcohol exposure is something that generally occurs after development in adulthood, this variant is unlikely to be subject to canalization. Because the aim of this lecture is to introduce the general principle and the limitations of MR, I've focused on one type known as one or single sample MR. Other types of MR have been developed, most notably two sample MR, which uses two different study samples to estimate the Z to X, or instrument to exposure, and Z to Y, instrument to outcome associations, and then uses this to estimate the causal effect of the exposure on the outcome. This can be useful when the exposure and the outcome are expensive to measure and when information on both isn't available in a single study. It also provides an opportunity to substantially increase statistical power by incorporating data from multiple sources, including large GWAS consortia. MR-EGR is a variation of MR that can be used when multiple genetic variants are used as the instrument. and it allows one or more of the genetic variants in an instrument to have pleiotropic effects. As mentioned earlier, bidirectional MR is useful for exploring the possibility of reverse causation, and it's essentially two MRs, one in the forward direction of X to Y and one in the reverse direction of Y to X, using separate instruments for X and Y. Intergenerational MR refers to MR studies of the effects of intergenerational associations, so for example maternal smoking during pregnancy and offspring IQ. There are a number of additional challenges and assumptions that should be considered, including the role of the offspring genome in mediating any effect of the parental genome on an offspring outcome. Intergenerational MR is usually carried out as a one-sample MR, but it can be conducted using two samples if a GWAS has been conducted separating out maternal and fetal genetic effects. As with all causal inference techniques, despite our best efforts, MR estimates are not likely to be completely immune from bias and confounding. Therefore, it's useful to compare evidence garnered using MR studies with evidence garnered using other approaches with different but unrelated biases. And this is known as triangulation. So to summarise this lecture, epidemiologists often have to rely on observational studies, and these can be limited in their ability to tell us about causal effects due to issues including confounding. Mendelian randomization uses genetic variants as instruments to study the effects of exposures or traits on outcomes in the presence of confounding. There are different variations of MR that can be used in different contexts depending on the research question and what data are available. MR makes several assumptions, some of which are difficult to assess, but there are strategies to mitigate some of the limitations of MR. Triangulating evidence garnered from MR with evidence from different approaches increases the confidence that observational associations represent true causal effects.